The topic of today's lecture is Introduction to the Autonomic Pharmacology, and conventionally the autonomic nervous system is part of the central nervous system, and central nervous system is part of the nervous system. So the nervous system is divided to the central and peripheral nerves. So the central nervous system consists of the brain and spinal cord, and the peripheral nervous system is located on the body organs and on the periphery actually. So this is the structure of the uh, nervous system, so central and peripheral nervous system. And then the peripheral nervous system is further divided to afferent and afferent divisions. And afferent is a sensory system, and the afferent division is divided further to the autonomic and somatic. So autonomic is mostly smooth muscles, and somatic is only the skeletal muscles. So, and then further the autonomic is divided to parasympathetic and sympathetic. So this is the structure of the nervous
Sir,
system.
wait a minute. So, as I know, the nerve system, the
The uh, the parasympathetic nervous system is mostly regulating the smooth muscles, cardiac muscles, and also gland cells, and it has preganglionic neuron. So preganglionic neurons body is in the central nervous system, and then the postganglionic neuron, the second neuron, and the acetylcholine is the neurotransmitter in the ganglia, so in this ganglionic cleft, and the receptor type is nicotinic. So while on the postganglionic neuron, the receptor is muscarinic, so the neuromediator is the same, acetylcholine, but the receptor is muscarinic, and this is a smooth muscle. Then the sympathetic is similar, also has two neurons, preganglionic and postganglionic, and the receptors and the neuron is the same in the preganglionic, but in postganglionic, actually the receptor is different. So most of the sympathetic, they have adrenaline, but sweat glands have a muscarinic receptor, the same as the parasympathetic. But most of the sympathetic mediated muscles have, so this actually the same cardiac smooth muscle and gland cells. Most of them have adrenergic neuron and the nicotinic, the alpha or beta receptor, and the neurotransmitter is norepinephrine. So this is a postganglionic neuron. And also there are renal vascular smooth muscle that are mediated by the dopaminergic receptors and with the dopaminergic neuron, and this is actually only on the renal vascular smooth muscle. Also, there is a adrenal medulla that is a gland that is also activated by the sympathetic nervous system, and the preganglionic neuron is one, and the second is not neuron, it's actually the hormone that is released to the blood, and then it's called the epinephrine, and then epinephrine will act on the adrenergic receptor on the different types of the muscles, the vascular muscles, and the cardiac mostly. So, and then there is also a somatic skeletal muscle that has only one neuron. So this is only one neuron, and the uh, the neuron is acetylcholinergic, and the receptor is nicotinic muscular type. So nicotinic muscular subtype. So these are the neurons and the receptors in the sympathetic and parasympathetic nervous systems. The um, the enteric nervous system has also its very complicated uh, structure, and the enteric nervous system is located in the walls of the GI system, and this is a simplified explanation of the enteric system, nervous system. So, like for a long time, we don't realize that there is our stomach and our intestine has a brain of their own. So that is why it's a very important system, because it will uh, make sure that you have enough food. So and the enteric nervous system uh, is a large and highly organized collection of neurons, and sometimes it's considered the third division of the autonomic nervous system, like sympathetic, parasympathetic, and then the third is the enteric nervous system, and it is found in the wall of the GI, from the esophagus to the distal colon, and it is involved in the motor and secretory actions of the GI. So the enteric nervous system includes the myentic plexus, the plexus of the Auchba, and the submucous plexus, the plexus of the Meissner. And the neuronal networks receive the preganglionic fibers from the parasympathetic system and postganglionic sympathetic axons. They also receive sensory input from within the wall of the gut. Fibers from the neuronal cell bodies and the plexus travel forward, backward, and in a circular direction in the smooth muscle of the gut to control the motility and the secretory cell in the mucosa. Sensory fibers transmit chemical and mechanical information from the mucosa and from stretch receptors to the motor neurons in the plexuses and the postganglionic neurons in the sympathetic ganglia. The parasympathetic and sympathetic fibers that synapse on enteric plexus neurons appear to play a modulatory role, all indicated by the observation that deprivation of input from both autonomic nervous system divisions does not abolish GI activity. In fact, selective denervation may result in greatly enhanced motor activity. The enteric nervous system functions in a semi-autonomous manner, utilizing input from the motor outflow of the autonomic nervous system for modulation of GI activity and sending sensory information back to the central nervous system. The enteric nervous system also provides the necessary synchronization of impulses that, for example, ensures forward, not backward propulsion of gut contents and relaxation of sphincters when the gut wall contracts. So that makes sure that whatever you eat will not go back and you will not vomit actually. The anatomy of autonomic synapses and junctions determines the localization of transmitter effects around nerve endings. Classic synapses such as the mammalian neuromuscular junction and most neuron-neuron synapses are relatively tight in that the nerve terminals in small buttons very close to the tissue innervated, so that the diffusion path from the nerve terminal to postsynaptic receptors is very short. The effects are thus relatively rapid and localized. In contrast, junctions between autonomic neuron terminals and effector cells like smooth muscles, cardiac muscle, and glands differ from synapses in that transmitter is often released from a chain of varicose sites in the postganglionic nerve fiber in the region of the smooth muscle cells rather than from buttons. And autonomic junctional clefts are wider than somatic synaptic clefts, and effects are thus slower in onset, and discharge of the single motor fiber often activates or inhibits many effector cells. So the uh, the autonomic nerves usually are based on the primary transmitter molecule, acetylcholine or norepinephrine. Like this is acetylcholine, so this is uh, nerve. If it releases acetylcholine at the end, then it's called acetylcholinergic, and this nerve is the norepinephrine. It releases norepinephrine at the end, so it is red, and that is why it's called adrenergic. And this is a dopaminergic because it releases dopamine at the end, then it is called dopaminergic. So a large number of peripheral autonomic nervous system fibers synthesize and release acetylcholine. They are cholinergic fibers, that is, they work by releasing acetylcholine. And these include all preganglionic efferent autonomic fibers like this one, this one, this one, this one, and this one. So preganglionic always acetylcholine, and the receptor is nicotinic. And also the somatic is also it has only one neuron, but it's also acetylcholine. And thus almost all afferent fibers leaving the central nervous system are cholinergic. In addition, most parasympathetic postganglionic and a few sympathetic preganglionic fibers are cholinergic. The significant number of parasympathetic postganglionic neurons utilizes nitric oxide or peptides as the primary transmitter or co-transmitters. Most postganglionic sympathetic fibers release norepinephrine, like this postganglionic is mostly norepinephrine. It also can release acetylcholine in sweat glands and the dopamine in the kidney, but mostly it is a norepinephrine. That they work by releasing norepinephrine or noradrenaline. These transmitter characteristics are sympathetic fibers release acetylcholine. And dopamine is a very important transmitter in the central nervous system, and there is evidence that it may be released by some peripheral sympathetic fibers like the renal vascular smooth muscle. Adrenal medullary cells, which are embryologically analogous to postganglionic sympathetic neurons, release a mixture of epinephrine and norepinephrine. Finally, most autonomic nerves also release several co-transmitter substances, and uh, these are not only one. So this is actually only the major neurotransmitter, primary transmitter, but they also can release others. 5 key features of neurotransmitter function provide potential targets for pharmacologic therapy: synthesis, storage, release, and termination of action of the transmitter, and receptor effects. So then let's discuss these processes, and we will start from the cholinergic transmission. The So the terminals and pericytes of cholinergic neurons contain large number of small membrane-bound vesicles concentrated near the synaptic portion of the cell membrane. So uh, this like this vesicle, and so this is a synapse. So this is a synaptic cleft. So and there are also large dense cord vesicles located far from the synaptic membrane. So like this one, for example. So this is actually close one, and this is actually farthest one. Whereas the smaller clear vesicles contain most of the acetylcholine, vesicles are initially synthesized in the neuron cell body and carried to the terminal by axonal transport. They may also be recycled several times within the terminal. Vesicles are provided with vesicle-associated membrane proteins. So these red membranes are vesicle-associated membrane proteins, and they serve to align them with the release sites on the inner neuronal cell membrane and participate in the triggering the release of the transmitter. So these vesicles actually will promote the release of the transmitter. The release site on the inner surface of the nerve terminal membrane contains synaptosomal nerve associated proteins, which interact with the vesicle associated membrane proteins. So these proteins interact with these proteins, and then that will lead to the release of the vesicle. Acetylcholine is synthesized in the cytoplasm from the acetyl coenzyme A, and the choline. So acetyl coenzyme A and choline are connected, and the catalytic action of the enzyme is required. The enzyme is uh, choline acetyltransferase. So this is choline acetyltransferase. This enzyme will catalyze the reaction. Acetyl coenzyme A is synthesized in mitochondria. So this comes from this is the mitochondria. So hope you studied the botany well. And the mitochondria are usually, they have not only one, they have many of them. So acetyl coenzyme A is synthesized in large amount of quantities. And choline is transported from the extracellular fluid by a sodium-dependent membrane choline transporter. So this is a choline transporter that will transport the choline inside. This transporter can be blocked by a group of research drugs called hemicholineums. So this is hemicholineums are the new drugs that actually can block this co-transport. So co-transport is with the sodium ions. Once synthesized, acetylcholine is transported from the cytoplasm into the vesicles by a vesicle associated transporter. So this is another transporter. There are many transporters here that is driven by the proton efflux. So the acetylcholine goes inside, the proton goes outside. So this is a proton efflux, and this antiporter can be blocked by the research drug besamicol. So this is another drug, besamicol, that inhibits this transport. So here it was a co-transport. This is the antiporter. Acetylcholine synthesis is a rapid process capable of supporting a very high rate of transmitter release. Storage of acetylcholine is accomplished by the packaging of quanta of acetylcholine molecules. Usually it's about 5,000 molecules in each vesicle. So quanta is the amount of the molecules in the vesicle. At most of the vesicular acetylcholine is bound to the negatively charged vesicular protoglycan. So vesicular protoglycan actually keeps it inside. So vesicles are concentrated on the inner surface of the nerve terminal facing the synapse through the interaction of so-called snare proteins on the vesicle, and snare is actually a subgroup of the vesicle associated motion proteins, and on the inside of the terminal cell membrane there are also snaps. So these actually are the snare proteins. So Physiologic release of transmitter from the vesicles is dependent on extracellular calcium and occurs with when an action potential reaches the terminal and triggers sufficient influx of calcium ions via N-type uh, calcium channel. So calcium goes inside and that triggers the uh, release of the vesicle. Calcium interaction with vesicle associated uh, movement proteins, synaptotagmin on the vesicle membrane and triggers fusion of the vesicle membrane with the terminal membrane and opening of the pore into the synapse. The opening of the pore and inrush of uh, cations results in release of the acetylcholine from the proteoglycan and exocytic expulsion into the synaptic cleft. So acetylcholine is in the synaptic cleft now. One depolarization of a somatic motor nerve may release several hundreds quota into the synaptic cleft, that is about 5 million molecules. One depolarization of an autonomic postganglionic nerve, varicosity or terminal, probably releases less and releases it over a large area. In addition to acetylcholine, several co-transmitters are released at the same time, and the acetylcholine vesicle release process is blocked by the botulinum toxin. So this is a botulinum toxin that will block the release of the acetylcholine through the enzymatic removal of two amino acids from one or more of the fusion proteins. And after release from the presynaptic terminal, acetylcholine molecules may bind to and activate an acetylcholine receptor, that is a cholinergic, one of these green ones. Eventually and usually very rapidly, all of their acetylcholine release diffuses within range of an acetylcholine esterase. So this is another enzyme that will split the acetylcholine into choline and acetate. So choline is actually goes back to the neuron later, and neither of which has significant transmitter effect, not choline, not acetate, and um, thereby this terminates the action of the transmitter. Most cholinergic synapses are richly supplied with acetylcholine esterase. The half-life of the acetylcholine molecules in the synapse is therefore very short, so about half of a second. And acetylcholine esterase is also found in other tissues, for example, red blood cells and other cholinesterases with a lower specificity for acetylcholine, including the butyrylcholine esterase or pseudocholine esterase are found in blood plasma, liver, glia, and many other tissues. So I think we can stop here and we will continue on next time.
Okay.
Do you have any questions on this?
No specific questions.
No, everything is clear.
Everything clear? Baha,
Hey. Yes, yes, clear.
do you had questions about the drugs in your country?
My question was, what specific drugs we use to the central nervous system? Is it different between the Russian and other countries? Because usually Russian they have their own treatment.
No, the treatment is the same for all humans. So this the structure of the the nervous system is the same in all animals, not only humans, and also most of the drugs that are used for humans are also can be used in animals, like cats and dogs.
Understand.
So if no questions, then thank you for your attention, and see you next Thursday.
Thank you. Have a good day.
Have a good day
Thank you.
too.
See you too.
See you.
Goodbye.
Bye bye.